is Dr. Tamás. He is a molecular biologist from the Institute of Clinical Experimental Research, and he is going to speak about modulated electro-hyperthermia and combined primary immortalized NK-cell therapy in human A2058 xenograft model. Thank you.
Thank you very much for introducing. Dear audience, I would like to show the extension of our study, which was presented in the last year Congress. It's about how to combine the NK-cell therapy with the modulated electro-hyperthermia on a human A2058 xenograft model. It's an experimental in vivo model, and we wanted to see if the electro-hyperthermia can make a favorable microenvironment for the NK-cell trafficking. So, if it would, okay. So, I think that the medical doctors in the audience might know it much more better than me, that the melanoma has really poor survival rates based on the CE reports. And nowadays, it's not a secret that the melanoma has so much immunosuppressive mechanism, which can be inhibited by the first-line therapies, such as PD-L1 inhibition or the CTLA-4 inhibition. And as we know, that the modulated electro-hyperthermia can induce some kind of adaptive immune response in the treated tumors. But so far, we haven't tried to complement this complement the electro-hyperthermia with immunotherapy in experimental models on melanoma cell lines. So we had a study which was published 2 years before. Sorry, just 1 year before, in the International Journal of Molecular Medicine, where we saw on B16F10 melanoma allografts that after the electro-hyperthermia, the size of the tumors reduced significantly, but we couldn't see any relevant increase in the CD8 or CD4 positive immune cells, which are the well-known participants of the adaptive immune system. But we saw that the MHC-I was reduced after the treatments. This can be a cause for the reduction of the adaptive immune cells, but the NK cells, the amount of the NK cells, tumor-infiltrating NK cells, wasn't changed relevantly. So we had the impression that maybe those cells were active, since they can be activated by the reduced MHC-I level on the tumor cells. So in this study, we hypothesized that after the electro-hyperthermia treatment, the MHC-I can be reduced on the tumor cells, and this can attract the NK cells if we add them from an outer source. So our model, so since we cannot gain enough NK cells from the mice, we changed the model for human melanoma cell line to can use the human NK cells as an effectors. So our model was the A2058 human melanoma cell line, which has a function of 53, and we injected them symmetrically on both femoral flanks of NOD-SCID immunodeficient mice, and then used the 30 minutes 42 centigrade modulated electro-hyperthermia, and the next day we injected on the lumbar region, subcutaneously on the lumbar region of the mice, the fluorescently labeled NK cells, which enabled us to follow the trafficking of them on an in vivo imaging chamber. And after this, we also monitored the tumor growth by ultrasonic measurement and checked the remarkable immunohistochemical markers, which was used for identifying the effects of the electro-hyperthermia. First, before we started, we tried out two kinds of electro-hyperthermia protocol. One was just a one-shot treatment, and on another way, we used the three-shot treatment. So here is the three-shot, and here is the one-shot treatment. These two end up, it was good for checking the electro-hyperthermia efficacy, and after that we identified that the only one-shot treatment was enough to reach significant tumor growth inhibition, we used that. But before that, I would like to show that how the two kinds of treatment acted. So after one-shot treatment, we also can show the significant increase of the cleaved caspase-3 level, and after the three-shot treatment, we also saw that on the treated tumor sites, and proving the apoptosis or the termination of the apoptosis, we could show out the double-strand breaks by the histone 2AX protein. It was also significant in case of the one-shot treatment on the treated side, and also significant on the treated, but also on the untreated side after 3 times treated tumors. So, and the tumor growth, so when the tumor growth curves gave us quite nice results, because after the one-shot treatment, we could see the significant reduction of the tumor sizes after 3 and after 4 days compared to the untreated side of the electro-hyperthermia treated mice, and compared to the sham-treated control mice. And after the 3-shot treatment, we also saw on the 3rd day the significant, on the 4th day, the significant reduction in the tumor size, which was maintained after that. But after this experiment, we decided to use the one-shot treatment because it's enough to check that. So 24 hours after the electro-hyperthermia, we could show out the significantly larger dead or damaged area on the treated tumors. And in context of this, we saw the relevant expression or increasing expression of the HSP70 heat shock protein. And furthermore, we saw the p53 activation was also higher after the electro-hyperthermia treatment. And it might be a connection because of the p53 can induce the caspase-dependent apoptosis as well. And we saw the caspase-3, the cleaved caspase-3 elevation on these tumors. So, furthermore, we checked that what kind of changes can be done on the tumor microenvironment, which can positively affect the NK-cell trafficking. And we checked the matrix metalloprotease 2 expression, where we saw that in the treated tumors, it was significantly higher than in the untreated tumors, and even more in the untreated tumors on the dead area, the expression was much more lower. For checking the relevant cytokine expression, which can attract the NK cells, first we established an in vitro model, and here we can see the relevant increase in the apoptotic cell fraction, and we checked three kinds of CXCL ligand, which can attract the NK cells in normal in vivo environment, and furthermore, these cytokines can be promoted by the heat shock. So we were curious how it goes, and surprisingly, the CXCL 9 and 10 levels was reduced after the electro-hyperthermia, but the CXCL 11 was significantly higher in the treated cell cultures. For the NK cell therapy, we use both primary human NK cells, which were isolated from a human peripheral blood sample, and we use the commercially available NK92MI cell line, which was a present from Dr. Samuel Hisham Wang, the former presenter. And for testing the killing ability of these cell lines, we checked the in vitro killing of the A2058 melanoma cells, and after we tried out that what they gonna do in the in vivo environment on our xenograft model. So in vitro, we could see that raising the cell number of the NK cells can cause the cell killing effect at 37 centigrade as well. But when we applied the 42 centigrade just by water bath, we could see a higher killing efficacy between the 37 and 42 centigrade cells. For this method, we used we loaded the cells with fluorescent molecule, which can release upon the cell damage. And I just would like to show how can it be visualized. For example, by microscopy, by phase contrast microscopy, we can see that after 2 hours culturing with the highest number of cells and the highest number of NK cells, they maybe around 70% of the cells, 70% of the tumor cells are died. And in case when we fill up these cells or with the fluorescent dye, which can be released upon the cytotoxic killing, we can see this kind of effects. So the NK cells are not labeled, but these are the tumor cells. And if you see the highlighted cells with the arrowheads, you can see that in the end of the movie, they just shrink because of the effect of the NK cells. And if you can see somewhere the NK cells are just the shadows of them just going through on the cells and attacking the tumor cells. So we made the xenograft model, and it was the 4-day protocol. At the day 0, we applied the electro-hyperthermia, and on day 1, we injected the NK cells, both the NK cell line or the primary. And after that, we monitored the increase or the trafficking of the NK cells. Since they are labeled fluorescently, we could see if they go to the to the electro-hyperthermia treated side tumor. And this was also proved by excision of the tumors, and we can see on the ex vivo tumors the accumulation of the NK cell fluorescent dye. And we could also show out the NK cells in situ in the tumor sections by the CD57 marker. And here are the numbers for the fluorescent measurement of the NK cell trafficking by the in vivo chamber. And here we can see that both the primary and both the cell line, the NK cell line, was significantly migrated more. The migration of the NK cell lines were significantly higher to the treated tumor sites. When we compared the damaged tumor areas, we could see also a slight difference between the electro-hyperthermia treated and the combinated treated tumors. Although it's not significant, we think that it's remarkable. And one interesting thing, which is also not significant, but we have a strong suggestion that in case of the electro-hyperthermia and NK cell combination, we also see a slight elevation on the untreated side. And we are quite curious on this kind of abscopal effect. And maybe later we try to find out if it can be significant or not, or what's the mechanism of that. So discussing all our results, we saw that the A2058 xenograft model, on this model, the electro-hyperthermia could induce the p53 mediated, maybe the p53 mediated tumor growth inhibition and caspase-dependent apoptosis. And we also saw the elevation of the matrix metalloprotease enzyme and the CXCL11 production in the tumors, which can prove or provide favorable microenvironment for the NK-cell trafficking. And furthermore, we saw that these NK-cells that we used, they are able to kill this tumor type, and in vivo environment we could see the significant migration of the NK-cells, both the primary and the cell line, towards the electro-hyperthermia treated tumors. But of course, further testing is needed to see if we can find an abscopal effect, or to see what's the basic mechanism of this attraction. So thank you for your kind attention.
Oh, thank you so much. It's really, really interesting, because I think the non-thermal effects of electromodulated hyperthermia are really interesting. And from my point of view, doing this combination with dendritic cells, or putting some cells inside the tumor and the hyperthermia treatment, it looks like very promising. So do you have like any idea if you will be able to begin something with humans in a short time frame, or something like that?
As I know that the NK-cell therapy is not as frequently used in the human clinics. So if I remember well, maybe it's mostly used in private clinics. It's not suggested that the first one between the first line therapies. But I think maybe it can be a little bit better than the cytokine therapies, because you might know that they hasn't got so much effect. And complementing the cytokine therapies with the adaptive NK-cell, or adding NK-cells, maybe derived from healthy human patients, I think it can be useful, because you know the NK-cells also, the NK-cell therapy hasn't got so much side effect because of the short lifespan of this cell type, and since they have this ability to attack the cells which don't express the main histocompatibility complex, and the tumors sometimes try to hide away from the immune system. Maybe I think it can be useful after much research.
Yes, I think it can be interesting also in those patients where we don't have treatment at all. So those bad cases but the problem is usually that is a very expensive treatment nowadays. As you said, there are some things in different private centers, but many times patients cannot afford paying that. But I hope soon we can have a trial or anything, because I think it's really, really very interesting. So thank you for your research.